Fine, great. So, hello, hi everyone. So, thanks for registering this particular webinar. Named as Exploratory Data Analysis for the Biomedical Datasets. Right. So, in this particular webinar today, which is of duration 1.5 to 2 hours, basically, I will try my best to make you understand the importance of exploratory data analysis. What are the different components which are required in this particular field? Right. What are the different fundamentals which you, being a student, we need to understand? What are the different sources from where we get the omics data? And what is really the meaning of omics basically? Right. So these are the few fundamental things that we'll be trying to understand by the end of today's session. As well, I'll be trying to give you a few hands-on session as well in Python so that you'll be trying to understand and correlate that how to make use of programming in the today's world for data analysis. Right, because if you will start go and apply for some internship, for some jobs or something, everybody nowadays want a particular biomedical student to understand programming. They want a particular student to do some analysis. They should be able to perform experiment, get the data, and do analyze the data on their own and get the findings from there. Right. So in the today's world, data analysis has really become a very important part of biomedical sciences. Right. So this is the motto of today's session that I'll be trying to make you understand. And this particular webinar is the first webinar of this webinar series, basically named as Omics Data Science. Right. So Omics Data Science is a course also that we are going to begin from 15th of April, basically. It's quite possible that we might delay for some more time, basically, because the webinar will end on 20th. Right. So it will be a long, detailed course. And few of those sessions from that course, I'm just trying to bring in before you through these webinars. Right. And the next webinar will be on 9th of April, that is predictive modeling for transcriptome datasets, right? So without any further delay, let us start the session. Great, guys. So see, so the first question that we should ask basically is that because we are spending our 2 hours of time, I'm spending my 2 hours of time and you're also giving your 2 hours of time. You should be able to understand that this time should be valuable. You should not waste your time, right? So by the end of this session, you will be able to understand what is the meaning of single omics and multi-omics, right? What are the different kind of the data sets in the field of omics? Why multi-omics is preferred over single omics nowadays, right? And the second question with that will be understand is that why multi-omics based signature discovery is required. Okay, because we know that biology doesn't work in silos. It doesn't work in isolation, basically, right? Each and everything is related. And this is what is known as the system biology, right? We know that gene leads to the DNA leads to RNA leads to protein. And in that process also, a lot of epigenetic changes and environmental factors and other things plays their role. Right? So how we can understand the biology at a system level? Okay. So in that direction, multi-omics is nowadays preferred a lot. Okay. Then we are looking at a word as signature. So then we were trying to understand what do we really mean by signature? Right? What is the meaning of signature in the world of biomedical sciences? Okay. Then we were trying to understand the role of healthcare data science in the 21st century and its scope. Trust me, guys, India is one of the country having the great potential for data science in field of healthcare, right? Because we are the country having the world's maximum population. And we also know that we're just a developing country. So the burden upon the shoulders of the doctors is very high, right? So AI data science has a great scope in that particular direction to help the doctors to make their life easy and make the lives better of the patients as well, right? So we really need to understand why healthcare data science and what are the different scopes in this particular domain. Then statistical inference and descriptive analysis with the help of Python. So by the end of today's session, I'll just try to understand, touch upon these topics and make you familiar with these concepts, right? But before that, So when I was going through the number of registrations basically, and the students and their background, I found that the students come from the first year of bachelor's to the PhD level as well, basically. Right. So I'll be trying to cover from the scratch and go to the higher level, basically. Okay. But still, even that, if any of you have any doubt in between at any one moment of time, feel free to unmute and ask a question. I'll be more than happy to answer your queries. And if I do not know the answer, I will try my best, look into the answer and get back to you. Okay, fine. So first of all, now let us try to understand what do we mean by health and the data, right? And what are the different kind of the data that we get. So let us try to make the session a little bit interactive. So will any of you would like to unmute and speak? Like, what do you understand by health, by the way? Okay. What do you mean by healthy condition? Hello, hi. Anybody would like to answer? Hello.
Feeling physically and mentally good.
Okay, feeling physically and mentally good. Nice. That's a right answer. Yes. Physically, mentally. One component you have missed. That is socially, right? So we should be socially, mentally, and physically fit. So any individual who is socially, mentally, and physically fit is considered to be healthy, right? Now, when we look at these different verticals of the society and the health definition, basically, we really need to ask, are we really healthy nowadays? Right? We are in the era of AI. Okay, like you just search something over the Google and your Instagram and YouTube and other things start recommending like recommend related products into your feed, right? You just try like you look for a particular song of your interest in the YouTube and it starts recommending the similar songs to you, राइट right? आप एक प्रोडक्ट सर्च करते हो Amazon के ऊपर उसके जैसे 10 प्रोडक्ट आपके फीड में आ जाते हैं यू शुड ऑल्सो गो फॉर दिस प्रोडक्ट इट माइट बी ऑफ इंटरेस्ट सो एआई has created literally a great place in everybody's life nowadays basically, right? Then when you start scrolling through your Instagram आपने देखा होगा like everybody nowadays is great fan of reels एंड अब आप एक बार जब ऑन करते हो आपको पता भी नहीं चलता है एंड आप अपने आधे घंटे एक घंटे कैसे स्पेंड करके खत्म कर देते हो रील्स पे राइट इट इज अ फैक्ट राइट when you will go and talk to some psychologist some doctors they will tell you that in the last 4 to 5 years the time span of the students the retention time span of the students and the people have really gone very down okay because these things रिटेंशन टाइम आपका बहुत कम हो जाता है लाइक फोकस टाइम आप फोकस नहीं कर पाओगे बहुत चीजों पे राइट सो reels are really like a great way to say a lot of things in a very small amount of time but when what kind of the product should be there okay what kind of the things should be made this we really need to look upon right because our concentration time span is really decreasing, which is a great concern for the doctors nowadays, right? Similarly, when we talk about mental health, we know that every year, like a number of students commit suicide due to academic pressure. Then so many people commit suicide due to financial issues, right? Number of people lose their lives due to some diseases and all, right? So we are not even great healthy at the mental level, right? And definitely at the physical level. If you will go back and try to look for some literature, you will find that post 2010, the number of diabetes patients has really increased a lot, right? And why, when you will try to understand the why part of this question, basically, you'll find a lot of reasons there. And definitely AI has a great role in that as well. Okay. So never ever assume that AI only has the pros, it has a great number of cons as well. And it is affecting your health, right, guys? But yes, uh, we need to make the lives better of the patients and the society. then we really need to make use of AI in nowadays, basically, right? With this, we really need to understand now what is the meaning of data, because this is what we have talked about the health, right? Then we really need to talk about the data as well, like what are the different kinds of the data that we get from there, right? So when you talk about the health and the diseases, okay, health and the disease, like here you can see number of health and the disease conditions, like see diabetes, okay, cardiovascular diseases, pregnancy. So no, pregnancy is not a disease basically, but in the pregnancy we get the data. And during pregnancy, if the female is diabetic, then it leads to number of complications. If the female is hypertensive, it might lead to number of complications, right? And sedentary lifestyle is making you very unhealthy, right? To make ourselves better, we need to do some exercise. We are gaining weight because we are not doing exercises. Then cancer is one of the disease and so on. So here we are just showing few of the health conditions and the diseases basically, right? From there, we get the data basically. What are the different kind of the data sets that we get? Okay. So when you look upon the data, like if you look into the biology, right? So when you look try to understand the biology at the system level, basically, you will get to find that this is the human in the center, and these are different kind of the omics data, basically, like genome, epigenome, transcriptome, proteome, imaging data, metabolome, lipidome, cytokines, right? So let us try to understand each of one of them one by one, basically, right? So see genome. Genome में क्या होता है आपके, like genome is made up of two words, gene plus O. So it's a group of the all the complete your genetic makeup of your body, basically, right? For example, we know that humans or the Homo sapiens are diploid organisms, right? So when you take the complete one set haploid set of the gene of the humans, it is named as the genome, basically. Okay. So when we try to understand and explore what are the different kind of variations which are happening at the disease level across the populations, okay, because we know that, like, if you will try to understand the breast cancer in the human Indian population and the breast cancer in the US population, you will find a lot of changes. These are because of the demography changes, right? And because demography affects genetics at a greater level, right? Then we understand epigenome. Epigenome is what? Epi means above, genome we have already looked at. Like, so anything which is not at the genetic level, okay, which is not directly affecting at the gene level, basically. For example, methylation, okay, then acetylation, salicylation, a number of PTMs, basically, post-translational modifications, and other epigenetic changes takes place. Okay. Transcriptome data is very simple, like the mRNA level data, basically. The RNA level data is named as the transcriptome. So genome is the DNA level data, transcriptome is the RNA level data. Then we have the proteome. Proteome is what? Protein level data. Sorry. Then we have the metabolome. Metabolome is what, basically? The metabolites, the different metabolites, which like the different entities which are there in your body, basically biomolecules, when gets processed, they gets reduced to the metabolite level, basically, right? So the complete set of those metabolite molecules named as the metabolome. Then in the lipidome. Lipidome is what? The complete collection of the lipid molecule and the cytokines. Okay. So these are at the molecular level concept, basically. And trust me, guys, it is only these things which regulates you, right? Suppose that I'm going to ask you a question. Suppose that आपके exam में question आया, okay, and you're not able to answer that question. And the question was at some chemistry level, basically. Okay. So what you really need to do basically in that situation is that you should just try to place yourself at that particular place of chemical. Okay. And just try to think, if you are stuck in that particular place of the molecule, what will you do in that situation? Trust me, whatever you will do in that situation, the same thing will that particular molecule will do as well. Okay. Because we as a human do nothing. Okay? It is all these molecules which makes you do something, basically. Right. So we really need to understand the role of the chemicals, the role of the biomolecules, but it is the molecules which are making me to speak right now. Trust me, I'm not speaking anything. It is the molecules which are guiding me to speak anything right now. Okay. If those molecules gets disbalanced or irregulated, I will not be able to speak a single thing. Right. So we really need to respect those molecules and understand the biology at the system level, basically. Got it? Great. So when we understand these at the bio level, basically, we really need to look at the different data sets as well. So for example, we get the EHR, electronic health record data. Okay. We get the social graph informations. Okay. For example, like we have the Instagram or in LinkedIn, basically, like everybody, like suppose that I'm connected to 10 people, then those 10 people are again connected to 10 people and so on. So I have a connection to the nearest neighbors of me, then to the nearest neighbor of them. Okay. When you try to understand and analyze those kind of the data sets, it is called as a social graph, basically. Okay. Wearable devices. So we are getting the data from the wearable devices. We get the data from the social graph, LinkedIn, Facebook data, okay, Twitter data, and all. Diet informations. Okay. Exposomes, microbiome data, basically, imaging. Okay, suppose that you are going, you are traveling by some car or some scooty, and you fell and made an accident. Okay, and there are the suspect that like you might have made some you might have made some fracture in your leg. डॉक्टर क्या करेगा आपका सिंपल लेग का एक स्कैन मार देगा स्कैन करेगा राइट एक्सरे so and x-ray is what it is going to give you a film we get a radio film basically right and what is the radio film it's nothing but simply an image what is that image made up of numbers okay because when you will try to understand the any scan image basically these images are nothing but simply images guys and these images are also made up of numbers okay because if I will show you today like how do a look like I'm right now looking to you in this particular format like a man with two hands with a mouth two nose and all sorry two ears and all right but in reality it is nothing but simply number computer को जब समझाओगे this will just be in the format of numbers and it will be just a matrix basically right so there are the different kind of the data sets that we get right now the thing comes to mind is that these are the different kind of the data sets that we get and this is the person whose health we really need to understand and make the lives better then we really need to ask a question or think of what are the different kind of the challenges and the questions that can be answered for example suppose that this particular person got a suffered from breast cancer so what are the few questions that you will ask right the first question will be that what are the molecules which is making this particular patient cancerous? Right. Another question will be that what led that particular molecule to change from the normal state to the cancerous state? Another question will be what are the different chemical molecules or the drugs which we can use to treat that particular patient? Right. So these are the different kind of the questions which we really need to ask, and we really ask these are the questions which we really need to answer. And in order to answer those questions, we make use of number of different kind of the analytical methods. And these analytical methods come from the field of data sciences and machine learning, basically, and simple linear algebra, right? Statistics. Okay. So there is one field of multi-omics basically where we are not going to analyze these data sets in silos. In silos means not in isolation. One possibility is what? You are going to analyze the genome data separately, epigenome data separately, transcriptome data separately, and no data are talking each other basically. Okay. Another possibility is what? You're going to take into account the interaction of each of these data sets together and then trying to analyze the data set and finding in meanings and inferences from there. So when you try to integrate the different data sets together, it is termed as multi-omics. When you try to analyze the different data sets in isolation, it is termed as single omics, right? So these are the different kinds of the methods for integration methods. I will not be touching upon that. But yes, there are three different kinds of the possible methods: DL based, deep learning based, supervised machine learning based methods, and unsupervised machine learning based methods. Okay. For example, like suppose that if you really have to ask a question, then the question can be like, you need to find the disease risk prediction, that is prognostication. What is the possibility that based upon the condition of these molecules, the particular patient is going to suffer from breast cancer in the coming 5 years down the time, right? Or you are given the gene expression data of the patients, and now you are asked to stratify the patients into different classes based on the expression value, right? You are given the patients and the two different conditions, the healthy and the control, healthy and the cancerous patient. And now you need to find out what are those molecules which are making a patient cancerous, right? Those set of the molecules which make a particular patient cancerous from the healthy conditions, or the molecules which help you to discriminate between the two given biological conditions, is named as biomarkers. And the journey of finding out those biomarkers named as biomarker discovery, basically. Okay. And then we have the molecular insight. So definitely, like when you are analyzing these data sets, you really need to go deeper and understand how the different molecules are talking with each other, affecting each other, and playing a role in signature discovery, basically. Right? How we can make use of those molecules, go for drug identification, drug repurposing purposes, and all. Okay, guys. So hope this particular slide is clear to all of you. If any of you have any question or doubt here, you can ask me here. Anybody have any doubt or something? Hello, guys. Am I audible now?
Yes, sir, you are audible.
So good. So great. If no question, then we can proceed further. Great. So now you can see here, guys. Here I have posted here like basically like omics and its type. And the question is that what do we mean by omics? And why are they important, first of all? Right? So this particular image I have taken from one research paper. Okay, this is the research paper basically. So there are like this is a central dog, that is DNA codes for like DNA replicates to DNA. Then this DNA gets coded to the RNA, and RNA gets coded to the protein. And from there we go to the metabolite level, basically, right? So when we study the DNA at the DNA level, basically, it's named as the genomics. Okay. And genomic study helps you to identify genetic variants in the DNA sequence, including coding and the non-coding regions. Right? You know that a number of proteins are coding, a number of proteins are non-coding, right? Why? The question is why, basically. You need to ask, right? Then epigenomics, that is above the genomic level data. So it helps you to evaluate non-coding, non-DNA sequences changes such as methylation. So these are not going to happen at the DNA level, basically, in the DNA, basically, right? It is going to affect or act from outside, basically, right? So then we go to the RNA level. So this DNA then gets transcribed to the RNA. This process named as transcription. Okay. At the transcriptomic level, you're required to evaluate the evaluation of quantity and quality of the RNA, basically, right? So definitely, like what is the quantity in which a particular gene is expressing, right? Because we know that, suppose that we already know that BRCA1 and BRCA2 are the biomarkers in breast cancer, right? So what is the level of expression of BRCA1 and BRCA2 in other diseases? Okay. Or suppose other gene like FOXA1 I'm interested in. So I'm asking question, what is the level of expression of FOXA1 gene in breast cancer? Right. So when you try to quantitate, the level of the expression of the transcripts, it is named as, all these things are done in the transcriptomic study, basically. Then we have the protein level data, basically. right? So from the protein, we go to the proteomic level. See, the DNA codes for RNA, but it is never true guaranteed that if RNA has been made, then it will definitely be translated to the protein. It is not definitely true. It's quite possible that in that process, some microRNA affected and got degraded, or due to methylation, it was not able to express properly at the protein level, right? So then we really need to quantitate and understand either a particular gene was able to translate properly or not, right? So when we try to quantitate the expression of the protein gene to the protein level, it is named as proteomics basically. So it helps you to evaluate a protein map network. So definitely when we get the proteins, we try to build a protein map network and understand how the different proteins are interacting with each other basically, right? And then we have the metabolomics. So it helps you to evaluation of endogenous and exogenous metabolites basically. Okay. So in the metabolomics, multi-omic data basically, see like what do we mean by multi-omics data mean, right? So see like these are the different kinds of the omics that I've shown here: genomics, epigenomics, transcriptomics, proteomics, metabolomics, and phenomics. Phenomics are at the phenotype level data basically. Okay. Then sorry, like I need to remove it here. We're fine. Then we have the clinical data basically, right? So phenotype level data, you also get the clinical data, radiomics data, and other screening based data. So these are the omics level data basically, molecular biology level data. They are at the clinical level data basically, right? Then we try to integrate these different data sets together, and we can perform either clustering, we can do either prediction. Okay, we get the different features from there basically. Okay, so for example, we have the genome, epigenome, transcriptome, proteome, metabolites. We are interested in to find smaller set of the molecules with the help of which we can discriminate a particular patient from the other patient. Those set of the molecules which help you to discriminate a patient from the other patient or a condition from other condition is termed as biomolecule, is termed as features and the biomarkers, right? So, and these features, we then try to understand how these different features are interacting with each other. Okay, so that is really even very interesting. Okay, so you can see here the biomarkers. These biomarkers are helping you to cluster the patients. Okay, so these biomarkers help you to cluster the patients. These biomarkers help to build a multi-layer network and all. Right? This again picture I've taken from some paper basically. Okay. So hope I am able to convince you and not boring you here that why multi-omics data and single omics data is really important. What are the different kinds of the data sets that we get, right? Then we really need to ask a question. So now let me ask you guys here. So I would love you to like hear from you that what are the different kinds of questions that you can ask in this kind of situation basically. Okay, so let me ask somebody here. Hey, hi Aditi, are you there? Hello. Are you sure not there? Okay. Aisha, can you hear me? Hello.
Yes, I can hear you.
Great. So Aisha, can you think of like any one of the questions that you can answer in these kind of the conditions?
Uh, See, if we talk about metabolomics, transcriptomics, proteomics, generally omics-based study, then in that case, the data of these databases or the data sets we are going to use or we available for
Hmm.
analysis of these data can be useful for biomarker discovery, as you said, but
Hmm.
they can also be used in some other information also or in some other way also. So we can
Hmm.
think about that too.
यस डेफिनेटली तो अभी आप कोई एक क्वेश्चन बताओ यहाँ पे आयशा जो क्वेश्चन आप सोच पा रहे हो
Related to these
ट्रांसक्रिप्टोमिक्स
transcriptomics, what are the data sets we are going to use?
मान लो अगर ब्रेस्ट कैंसर और हेल्दी पेशेंट का डेटा क्या क्वेश्चन पूछोगे आप
If it is about breast cancer or what are the data, like what is the information we have for breast cancer patient or healthy patient? And
ठीक है,
another thing is I can ask how the data has been collected or how the data has been carried or you can say processed, I can ask.
great. So that is data processing level basically you can ask, right? Great.
Exactly.
So Anirudh, hi Anirudh, can you hear me?
Yes, sir.
Great. So Anirudh, can you add something here? Like what is the question that you would love to ask here?
One of the few questions I'd like to ask is, for example, we are doing a screening for a biomarker detection. How
Hmm.
could we find out the specificity and sensitivity based on a particular patient we are considering? And based on
Absolutely,
the
yes.
type of sample we are taking, whether it's blood sample or how are we exactly screening for a particular patient? And we can use that to basically use omics data analysis to find out which be best suited for a particular patient, particular sample, and our
Right.
target organ or what we're exactly thinking about.
Yes. Definitely great. So you mean to say basically that like we can try to find some markers, and with those help markers, like we can try to find the how sensitive or like how specific can we be in discriminating one disease from the other, right?
Prevent cross reactivity, which has the
Yes.
least cross reactivity based on different diseases or different cancers. For example, we're taking like breast cancer and colorectal cancer. If there is any
Right.
cross reactivity or which has the
Yes.
least cross reactivity, so that we can enhance our outcomes.
Right. Great. So hi Garima, are you there? Hi Garima, can you hear me? Hello. I guess she's not
Yes,
there.
sir.
Hello. Hi Garima, are you there? I is no. Hi Ritika, can you hear me? Hello. I guess people have joined and left, gone somewhere, right? Mitali, are you there? Hi, Mitali, are you there?
Yes, sir, I'm
Tanishri,
there.
yeah, great. So, can you try to answer, like, think of something like what is the question that you'd like to ask here? Any question?
Yes, sir. Specifically, when we're like dealing with a cancer data set, like I would like to ask questions specifically related to answer which kind of like uh, the patient history they are dealing with, like the previous data that we are dealing with. So that kind of questions I'd like to ask, and the type of reactions or the kind of medication that they are undergoing and what kind of reaction is shown in them, that kind of questions I can ask.
Okay, great. So, it means like relating the patient level, the clinical level data with the transcriptome level data, right?
Yes, sir.
Great. So, let us see like what are the different questions that I have framed here. Okay, so now see here basically. See. So first question can be like, what is the distribution of the expression level across all the genes? Okay. So, so what is the distribution of expression levels across all the genes basically, right? So suppose like we're talking like here, like suppose that we are taking only 10 genes. Okay. So what is the distribution of these 10 genes in the two different conditions, that how the genes are expressing in these in the given conditions basically. Okay. Another question can be like, which was answered by second person basically. That is like, or the first person, Aisha basically, that we can perform complete EDA. That is, we will try to explore the data. How many values are missing? How many values are zero? We're not able to find the data basically, right? Then we can ask the question like, which genes show the highest and the lowest expression levels, right? Which particular gene is having the highest expression value in the breast cancer or in the healthy patient, right? Another question where can be there. How does the expression vary across different samples or the condition? Okay, suppose that we are taking only one gene basically. Yeah. And suppose we are having total 10 different cases. 10 different type of the cancer basically. So you are interested to look at what is the, in which particular condition the expression of this gene is the highest and the lowest, how the distribution goes in these 10 different conditions for this particular gene basically. Then you can be interested to ask basically, are there outlier genes with extremely high or the low expression, right? So outliers are the samples basically. Okay, outlier are the samples which behaves orderly with respect to the normal samples basically, right? Then you can ask, what is the correlation between the expression profiles of the different samples? Do the certain genes cluster together based on the expression patterns? Then you'll be interested to ask, are there any batch effects or the technical variation between the samples? Or how many genes are differentially expressed between the conditions of the groups? What is the overall variability in gene expression across the data sets? So these are different kinds of questions that you can ask, right? And try to find from the given data basically. And this leads to the science of and application of statistics in the world of biomedical sciences, right? So here I'm just showing a particular few of the images which are used to answer these kind of the questions, right? So it is a histogram, it is a bar graph basically, a grouped bar graph or the stacked bar graph basically, right? Where we are trying to show the expression of few of the genes in the different conditions, the trio the different conditions basically, right? This is termed as the correlation heat map. Correlation heat map. That is, which shows us how the different genes are correlated with each other. Here we are showing a pie chart. Which is showing the proportion of the gene function categories or something basically, right? So we are showing just here the proportion of something basically in the different conditions, right? Similarly, to show the proportion, we can also make use of a stacked bar plot as well again. And then we are showing here a violin plot, show the distribution of the genes in that given condition. So you can see here, with the two conditions, healthy and the disease, right? So how the genes are expressing differently in the two different conditions. Right? So these kind of the questions we can definitely ask and answer with the help of these simple visualization methods, right? But in order to answer and understand these kind of things, the first question we really need to ask now is that, what do we mean by data visualization? Okay. So this was a task that was done by some scientists basically. Okay. So what we did basically is that like he asked few of the students to write about data visualization. What do they understand by data visualization, right? And then he created a word cloud. So here he showed the 150 words based on the 150 words. And here in the word cloud, the size of the letters is directly proportional to the frequency of the letters in the given corpus, right? So here 150 words we are showing. Here we are showing the 40 most frequent words. Here we are showing the top 5 words only, right? And now from these top 5 words, what you can see basically is that data, visual, way, information, story, right? So if I have to connect all these 5 words together, what I would find basically is that data visualization is nothing but simply a visual way to tell a story with the data and the information. Okay. And the goal of visualization is to aid our understanding of the data by leveraging the human visual system, highly tuned ability to see the patterns, right? Because we know that we being humans are very highly tuned towards the images, graphicals, as compared to the words, right? So this is the meaning of data visualization. That is telling a story through the data and building information, basically, right? So in field of biomedical sciences and bioinformatics, basically, this is really going to play a very important role, which I showed you in the previous slide as well, right? So now, when we talk about the data and all, basically, So then we really need to ask question that what do we mean by statistics? Okay, because all these kind of the questions basically, guys, that I have showed you here, these will be answered with the help of statistics, right? So statistics is nothing but simply it is a science whereby inferences about some specific random phenomena you're going to find from the limited sample material basically, right? For example, suppose that you have to understand the economy of India, then is it possible that you can take the income of each of the individual? It is not possible. You cannot go and ask the income of each of the 140 CR plus population people, right? It is not possible. So what we'll do in that case, we'll just try to pick some random individuals from each of the state of India, which will represent the population. Then we'll try to analyze those sample individuals which are representative of the target population. And then based upon those information that we have gathered and make the analysis, we'll try to make the inference about the population. Okay. And this science is named as statistical inference, right? And this is the great application of statistics. Okay. And in that process, we really need to understand two phenomena. Okay. No, one phenomena basically, not two basically, because second is the statistical inference itself. That is the descriptive statistics. Okay. And when we talk about the statistics, there are two different kinds of statistics, guys. One is the mathematical and other is the applied, right? So in mathematical statistics, we try to develop the methods for the statistical inference and the other analysis basically. Whereas in applied, we just try to make use of these statistical tools in the various domains to extract some meaningful inferences from there, right? And when we talk about the biostats, okay, so biostats is also what? It's a field of applied statistics where the domain understanding is going to be of the biological field, right? Great. So now here, I would love to now show you few of the analysis basically, but let me ask a question here first of all, that how many of you guys, just a minute, एक second, sorry. So let me see here. Docs. Where is the poll that I can create here? Reset, meeting info, whiteboard captions. Just a minute, okay? I'm just looking at the poll from where I can create. Great. So now I am going to share my screen basically. Please let me know if the screen is visible to you guys. Is this screen visible, guys? Hello.
Yes, it is visible.
Great. So see, guys, so those who are new here to the world of Python basically, so let me show you. So you need to just go to Google basically and just you need to type their collab. Okay. You just need to type their collab basically. Okay. And when you type collab there, you will get to find their Google collab. When you will type on Google collab basically, it will open something like this. Okay. And then if you have worked in the Google collab and made few of the files basically, it will come like this, right? And if it is for the first time for you, then you will go to file and you will get multiple options here, right? So from that, you will need to click on new notebook in the drive basically. Okay. And now it is opening a new notebook for me basically, right? So let me tell you what do we mean by Google Colab basically. So it's a platform that is offered by Google, okay, which lets you perform all the data analysis, data science kind of the work in Python basically. Okay. Now here you can do a number of tasks basically. What is the best part of Google Colab basically is that you can connect your drive directly with that, and you can access all the content from your drive directly to the Google Colab. Okay. Hanji, yes. Who's this? Anirudh.
Can
Please
you
go ahead.
can you please repeat how you set up the for people who haven't done it, Poland?
Google Colab.
Yes, sir.
Very simple. Like, see, once I'm repeating again, you just need to type here Colab on the Google. It will open. Then you need to click on the first link, Google Colab. Okay. इसे cancel कर दो आप, cancel. Okay. And then you can go to the file and then just need to click on the new notebook. Okay. Please make sure that for this you are logged in with your Gmail. Okay. Because it is directly going to be connected to your Gmail basically. Okay. So now you have opened a Google Colab basically. Fine. Now let me show you like what is Python and what are the different kind of things that you can do in Python. Very simple. So you can do very simple kind of the task like 2 plus 2. It will give you 4. We know already, right? You can type here print I am Alok Anand. Right? So I can type like this. I can try to few other kind of the complex jobs, like suppose that 1, 2, 3 and 4, 5, 6. And I need to add these two lists together basically. But I need to add 1 with 4, 5, 6, and add 2 with 4, 5, 6, and 3 with 4, 5, 6, right? So very simple. So for I in A, for J in B, and then I can type print I plus J. And now you can see here that 1 plus 4 is 5, 1 plus 5 is 6, 1 plus 6 is 7, 2 plus 4 is 6, 2 plus 5 is 7, 2 plus 6 is 8, 3 plus 4 7, 3 plus 5 8, 3 plus 6 is 9. Got it? So you can do these kind of the works. Also, let me show you what you can do basically here is that you can read any of the images here. Let me give you a few examples. So for example, this is my image here. It's me, right? And now I'm going to copy the path of my image. Okay. And I need to load my image and see here. So import matplotlib. Okay. And then I can read my image here. And now I will show you that. See, I was telling you like images are nothing but simply what? Numbers. pld.imshow. See guys, it's me, right? I told you that images are nothing but simply what? Numbers, right? So this is me, but what is this image really? It is what I am. Okay. Now what I can do basically is that I can look at
using the matplot ID, we have to install it or we can just use it.
Abu Dua repeat slowly, please. Yeah. Can you
So
have your
you
question
have
once
used
again?
the
Sir,
matplot
she's
ID.
Hello, आपकी आवाज कट रही है. Please थोड़ा एक बार और.
Sir, you have used the matplot ID for importing the image. So before
Yes.
using that, we have to install that, or is it already in the Python
No,
notebook?
like I will go there. Basically, let me show you. So here you have a folder sign. Basically, you will go there. Then here you have this button, right? So it is upload button basically. And then I can upload my image from my local computer there, right? Then I have copied the path of this. Okay. And first of all, I need to do this. I need to call a library here. That is matplotlib. Then there is a function named as imread. So I passed my path here, and then I have read the image here. Got it? Is it clear? Hello.
Sir, will
Sorry.
we be getting session recording for this?
So that I can not tell you right now. Okay. So that depends upon me. Either I will share the recording or not. Okay. Yeah. So fine, guys. See. So now this is the image that I'm telling you basically, right? So now this image I can show basically now. Like I'm more interested basically now that what is the dimension of this image? How many pixels are there? Because आप पढ़ते हो ना, लाइक आप अपना जब एक टीवी खरीद के लाते हो, आप उसको देखते हो लाइक ये इतने पिक्सल बाय इतने पिक्सल है। हम कहते हैं लाइक हमारी टीवी में ये इमेज ज्यादा अच्छी सुंदर दिख रही है, ये कम सुंदर दिख रही है, ऐसे क्यों? That depends upon the pixel basically, right? So I need to now look at my image. What is the dimension of my image? See, so my image is 1600 cross 900 cross 3. What is this 3? This 3 is for the 3 different channels. This is a RGB channel image basically, the red, green, and blue channel image. For that, we have 3. And these are the 2 dimensions, the length and the breadth dimension basically. That 1600 cross 900 number of pixels are there. That is what? 1600 cross 900, that is 144 tetra 0. These are the number of pixels that our image has. Higher the number of pixels, better the image will be there basically. Okay. Now suppose that you want to crop this image. Suppose that you want to crop this image. Okay. So 700 to 1200, then 600 to 800 and all. Okay. Now see, I've cropped my image, right? I have cropped my image. Okay. If you want to just look at the face, what I will do basically, see, it is 800 something and that, right? So let me show you the face basically only. 800 and okay, yeah. Sorry. Okay. And then here I need to show basically only how much. Ooh, all. ये देखो आप. Now you can see only my face. Okay. If I want to crop more area, I can do that as well. Okay. But here I can do like this only. Okay. There are different fields like image segmentation, image detection, image classification, object, and all basically, right? So what we do there, I can create a boundary along my image only this, like see, I will create a boundary only like this along my hairs, my nose, my ears, and all, and rest everything will disappear basically. I can do those. Okay. These are the things which we study in the field of computer vision in the advanced analysis method basically, right? That is none of our interest right now basically. Okay. So this is just an example like what are the different kind of the things that we can do in Python. Okay. So fine. So now let me come here basically. The slide that I need to cover basically for you guys. Okay. So see, I'm telling you that like you can mount your drive basically, and you can make use of the data from the server directly. See, not the server, but the drive. So what you need to do for that basically is that go to the files, this particular. Here you will find 4 different buttons. You just need to click on this third one, the folder button, mount drive. When you click on this, you will find something like this. Permit this notebook to access your Google Drive files, and you will connect to Google Drive. Okay. It is now mounting. It's mounting. It will take some time. Great. So I guess it has got mounted or not. Okay. No, I guess. Oh, it has got mounted basically. Now you can see here a drive button also on the top. Okay. Great. So now you can see here, I'm getting my drives, shared drives. Now I can go to my drive and I can access all my folders here in the chat. You see here, right? I can access everything. Okay, I can access everything. Let me show you some example, okay? So that you'll be able to appreciate these things. Is there some image workshop? Let me show you some example, okay? Fine. So I've copied the path, and then I need to basically load a library again, OS, and I need to now change my directory. What is the meaning of change of directory? Basically means that भाई अगर आप मुंबई में बैठे हो, ओके, और आपको कोई काम दिल्ली में करना है, इट इज नॉट पॉसिबल, आपको दिल्ली जाना पड़ेगा, राइट? वैसे ही आपका कंप्यूटर में होता है। अगर आपका कोई एक फाइल जो है पड़ा है डी ड्राइव में और आप बैठे हो सी ड्राइव में, यू कैन नॉट एक्सेस दैट फाइल डायरेक्टली, राइट? सो यू नीड टू चेंज द डायरेक्टरी फ्रॉम द डी टू द सी, देन यू कैन एक्सेस दैट। सो दिस इज व्हाट वी आर डूइंग हियर, change directory. And then I can list down all the files which are there. Oh, it is empty. It is telling me that it is empty. Not an issue.
Sir,
Yeah, please.
what is the work of OS library? Why it is
OS basically operating system. It helps you to like get the directory, get all the images, get all the content of that particular folder, to change the directory from one to the other, and remove the files, delete the files, and all those related works. Okay.
okay, sir?
Sir,
हाँ जी बताओ.
sir, as I use my Python through Jupyter Notebook, so can I there also upload my drive, or we can do only this through Google Colab?
In the local Jupyter, you cannot. Only drive Google Colab.
Okay. Okay.
So see, now I am loading, I am reading a file directory from my drive here. Like you see here, drive, my drive, workshop 2025, and this image, right? IMG.
Excuse me, sir.
हाँ जी बताइए.
So, what is the use in of this Python, like of this directory change in directory in the data analysis for biomedical data?
गुड, नाइस क्वेश्चन. अब देखो यार, ऐसा होता है, मान लो आपके पास दो डेटा, एक है क्लिनिकल डेटा, एक है ट्रांसक्रिप्टोम का डेटा, ठीक है? किसने पूछा क्वेश्चन? कैन यू टेल योर नेम?
Sir, Garima.
गरिमा, ग्रेट. सो सी, सपोज दैट गरिमा, आपके पास तीन डेटा है, ओके? एक है आपका ट्रांसक्रिप्टोम डेटा, एक है मेटाबोलाइट डेटा, एक है प्रोटीन डेटा, और एक है क्लिनिकल डेटा, मान लो, ठीक है? आपने ये चारों डेटा चार डिफरेंट फोल्डर में बना के रखे हुए हैं, ठीक है? गॉट द क्वेश्चन?
Okay, sir.
हेलो, ग्रेट. अब आपको क्या करना है? अब आपको मान लो ये चारों फाइल्स रीड करनी है और चारों की एनालिसिस करनी है. तो अब एक बात बताओ, आपने मान लो अभी आप जो लोकली हो, अगर आप लोकली मान लो ट्रांसक्रिप्टोम फोल्डर में बैठे हुए मतलब यू आर प्रेजेंट इनसाइड द ट्रांसक्रिप्टोम फोल्डर, कैन यू रीड अ फाइल ऑफ द मेटाबोलाइट फाइल देयर? No, it will
No,
not.
sir. No.
Right. So we need to do basically what? We need to change the directory first. Okay. We need to change the directory from the transcriptome to the metabolite folder, and then only we can read the files from there. Okay. This is one thing. Another option is what? You can directly give the complete path. If you can give the complete path, then it will read. Like you can see here, I'm giving the complete path. Like it is here, like content/drive. Within the content, I'm in the drive. Within the drive, I'm in the my drive. There are two drive options, my drive and the shared drives. Within that, I'm in the workshop folder. Within that, I'm in the 2025 folder. There, I'm having an image of mine. Okay. Now I can get my image like this. You can see here. And now you can see the dimension is 4000 cross 6000 here, guys. Let us see what sort of image is this. So plt.imshow. Hope I've answered your query, Garima, right?
Yes, sir.
Good.
Sir, one more question. Like
बस
I
एक सेकंड,
used
बस एक सेकंड. अब देखो ये इमेज आपको ऐसे दिख गई, ठीक है? ये इमेज की डायमेंशन आपकी जो है 4000 क्रॉस 6000 है. अब बता आपको क्वेश्चन क्या है?
So like I used Python by installing from the Google like Python. But like this is alternative or something else, collab. I
नहीं
don't know
नहीं,
about the
दिस इज
collab.
डेफिनेटली अल्टरनेटिव बेसिकली, गरिमा. ओके. इसकी ये है कि लाइक सबसे पहली बात ये है कि आपको कुछ भी काम सेव करने की टेंशन नहीं है, ये ऑटोमेटिकली आपकी ड्राइव पे सेव होता है, ठीक है? जीमेल पे आपके
Okay, sir.
ड्राइव पे सेव होता है. दूसरा ये है कि इसमें आपको स्टोरेज 100 GB रैम मिलता है बेसिकली. देन आपको टीपीयू भी मिलता है, इसमें जीपीयू भी मिलता है, ठीक है? मतलब आप बहुत ही हैवी कंप्यूटेशन कर सकते हो. अगला बेनिफिट क्या है बेसिकली कि एवरीथिंग लाइक यू कैन एक्सेस योर ड्राइव डायरेक्टली. अगर क्या है कि आपको बहुत बड़ी बड़ी फाइल्स है, आप उसको नॉर्मली लोकल पे कर लेते हो, बट अगर मान लो आपके कंप्यूटर में उतनी आपका रैम उतना अच्छा नहीं है, आपका मान लो कि आपका 2 GB रैम या 3 GB रैम कंप्यूटर है, आप नहीं कर पाओगे एनालाइज फाइल्स. Okay, so for that we need heavy computation power. So that is being offered by Colab free of cost. अगर आपको Colab में बहुत ही heavy computation चाहिए, then you need to purchase the server and pay them basically. Okay, so that is the benefit of Google Colab basically. Got it?
Yes, sir. Thank you, sir.
Good. So now you can see here, guys. I have read an image here. Okay. Now suppose that मैं दूसरा example दिखाता हूँ. So suppose that I am inside my folder. I am inside my this folder. Good. And I am going to copy my path here. So now I'll do what? os.chdr. And now you can see here, I have changed my directory here now. And now I'll just do it this os.listdir. And now you can see here that inside that I have number of files. Tanishka, Ingita, Himani, Simran, Sanya, Arti, all files and all. Now suppose that I want to go inside each of the folder and look at the content. Like what is the content of each of the folder? What I can do, see, let me show you. I will just copy the path here. I will call it as the parent path. Okay. And then I will do here os.listdir files. And what I'll do now here, I will change, because I told you, you cannot access the file inside Himani when you are inside this particular folder. So I need to change the directory here, right? So what I will do, os.path.join parent underscore path comma I for I in files file underscore path. Okay. And now let me show you file underscore path directory here. Now you can see here, I've got all the files here, right? So last one is not of my interest. So this is named as indexing. So I have selected all my file path except the last one, right? Now I'm going to access all my files, right? So now I'm interested in basically to look at the file path of each of them. So see what I will do basically. For I in file underscore path, what I'm doing, I'm going to access each of my file path. Then what I'll do, I will change my directory. Very simple. This I've explained you conceptually, right? Now I'm inside that file path. And then I will do what? os.listdir print my current working directory is this. Okay. Now see here. See guys. Okay. इसको मैंने प्रिंट नहीं दिया इसलिए इसने ऐसे दिया नहीं. Now I can see here guys. My current working directory is this. Content drive, my drive, academic counselor, Fab 2025 internship, Tanishka. Inside that, I am having these docs like BTech in genetic engineering .google doc, Google doc, and all basically. So by writing a simple command basically here, I can go and access each of the files directly. जो मुझे मैनुअली करना पड़ेगा, लाइक सपोज मुझे मैनुअली करना पड़ता बेसिकली कि मैं एक एक फाइल फोल्डर में जाऊं, मैं देखूं क्या कंटेंट है, मैं ऐसे एक बार में देख सकता हूं. एंड डू यू नो वन मोर थिंग, I can read this doc also here. I can read this doc also here in directly basically, but that I'm not going to do there, right guys? So similarly, you see here like my current working directory in this is this internship all files, and now here we are showing copy of BSc honor zoology, microbiology, micro copy task one TechMedBuddy, copy management is Google Doc and so on. So hope and see ये काम कैसे कर रहा है, बहुत सिंपल. आपका पहले पेरेंट पाथ था, अब जहां पे हो देन आपने क्या किया, उस पेरेंट पाथ के अंदर मल्टीपल फोल्डर्स हैं आपके, राइट? सो आपने उसकी डायरेक्टरी ले ली, फिर आपने पेरेंट पाथ में इसकी डायरेक्टली ऐड कर दी, डायरेक्टरी ऐड करी है os.path.join पेरेंट पाथ comma I for I in files, and then last one of was not of my interest, so I selected all but not the last. And now my task is to like go inside each of the folder and look at the content. What are the different files present there, right? So this I've done here basically. Very simple command. Got it? अब आप उसको ये करा कैसे देखो. So for I in file path. It means I entered inside my file path directory, which is a list. OS dot. So right now my first is what? First file path. os.chdir. So I'm changing my directory here now. And so I change हो गया. And now I'm writing, because you should know कर क्या रहे हैं हम. So my current working directory is this I. And then you're writing os.listdir. भैया मेरे सारी files दो क्या क्या files है. काम खत्म. Now if any of you have not understood, please ask me a question here. Hello. गरिमा आपको समझ में ये OS वगैरह सब क्या कर रहे हैं, कैसे कर रहे हैं?
Sir,
हाँ जी बताओ.
sir, if we, for example, we have two sets of data, for example, transcriptomics and metabolomics data, and we want to like analyze them and plot a graph to find out where exactly like there's a change with respect to normal cells and maybe disease cells. So how do we do that? By looking at the path or the
Good.
directory?
So that we will do in the second webinar basically. That is on the coming 13th, sorry 9th one basically. But I will show you few of those today basically. अभी मैं दिखाऊंगा वो आपको, ठीक है? Good guys. So after giving the brief overview of the Google Colab basically, now let us see what are we doing here. Like see. So first we are mounting a drive of mine. Okay. So here I'm going to mount my drive directly. So it is saying the drive is already mounted. Guys, you can see my screen now. Hello.
Yes, sir.
Yes, sir.
Good. So now you can see it like I'm going to load two my libraries, एक OS or एक pandas. Panda is a library which will help you to do all the data analysis basically here, right? Now you can see here os.change directory. So what I'm doing is simply going to this particular path. So content drive, my drive, workshop, DES dataset. So I'm going inside this particular directory. And now I need to look at what the different kinds of files present here. See, so that I've done here. So you see here, these are the different files present there, right? Gene expression data, lung adenocarcinoma.csv, lung adenocarcinoma clinical metadata.csv, and so on. From that, what I'm interested in basically is that these are transcriptome dataset basically, guys, that I've saved you for giving you a hands-on here. Like example, like what are the different kinds of analysis that you can do. Okay. So now in this, I'm going to make use of my TCL lung SAM HiSeq v2.gz file. Okay. So I'm going to read that file and show you how that file is going to look like. See. So see guys. So here we are showing you the first 10 lines of this file and how is it looking, but it's not looking that good because rendering is not great. See. So this is how our data is going to look like to you guys basically. Okay. So here is the gene name. Here is the gene name basically. This is the sample ID and this is the expression value. It means that in this particular patient, this gene is expressed with a value of 9.6503. Here, gene is RNF10. In this particular patient is expressed with this particular amount. Now you can see here that this gene is expressing in this patient at this much amount, whereas in this it is higher, whereas in this it is lower, right? Within the same patient, the gene was almost not expressed, which is RNF7, but RNF10 is present, right? So what set of the genes are expressing in each of the patient? So number of questions now you can ask guys here, okay? So let us do some EDA on this data set only first. Okay? So let me show you. So the first question is that I'll be interested to first of all understand one single gene. Okay, so let me see here first. So let me prepare the data a little bit more here. But this is how our gene expression data looks like. See, the genes and the patients we have. And these are the expression values, guys. Okay. Now we need to understand and have the clinical data also of these patients. Okay. So what are the different clinical symptoms of these patients? Okay. So that I'm going to read here. Okay. And now see, so I'll be interested to see what are the clinical features basically of the data. And now you can see here that these are the columns of the data which are having more than 4 unique values in that. Okay. So you can see here like EGFR mutation performed. Either EGFR mutation was performed or not. So there are 3 unique values, NAN, no or yes. Okay. Vital status of the patients, either living, deceased or none. Either the patient was living or the patient was died, or NAN values. It means some values we do not have. Then sample type ID. These are the different sample IDs, right? Sample type, see, primary tumor, solid tissue normal, NAN recurrent tumor. Radiation therapy, either radiotherapy was given or not. NAN, no, yes, discrepancy. These are the different type of clinical features available to you, right? From that, I'm interested in the primary disease basically. Now you can see here that there are 680 patients of lung adenocarcinoma and 619 patients of lung squamous cell carcinoma. Okay, so, and now you can see here that these are the patients, these are my genes, right? And now I'll do what? I need to add a column to the data which will tell that either this patient is adenocarcinomic or, sorry, squamous cell carcinoma basically, yeah. Squamous or adenocarcinoma, right? So for that, what I'll do, I will add a column in the last basically. So let me show you. See, now I have done this task basically. Okay. So it is writing basically. It is already written, but fine. It will get overwritten then. See guys, now here. Great. So can you see here now? These are the sample ID and here is the condition vector. Right? Now I'm telling that this sample ID belongs to the patient of lung adenocarcinoma. This sample ID belongs to the patient of lung squamous cell carcinoma and so on. Got it? Right? Now let us do some analysis. Okay. So see here, like what we are doing here. So now suppose we are interested in to understand about the HIF3A gene. So here HIF3A gene is here. And we want to understand its properties. We want to understand its property. What is the mean value of this gene in all the samples? What is the minimum value? What is the maximum value? What is the standard deviation and all, right? So let us see here. So there is a function that is gene exp, and then you are extracting this particular gene ID, and then you are putting dot describe basically. Dot describe will give you the complete property of this gene. And now you can see here that total number of samples 1129, mean value is 6.56, standard deviation is 2.39, minimum value is 0, and these are the different quartile values, and maximum expression value is 12.40. Okay, guys. So the range is what? 12.40 minus 0. So range is 12.40, right? Mean value is given to you. Median value is this. 50th percentile is the median value you know, right? Model value doesn't make any sense because it's a continuous data, right? Model value makes sense in terms of the discrete data basically, right? Got it here? Anybody any doubt? This is the one question that you can definitely ask, right? Got the clarity here, guys, or not? Hello?
Yes, sir.
Hello,
Yes.
good.
Yes.
Now let us see here. And the question will be that how many patients are the different conditions? So for that, you can do a function counter. And it will give you, see, 576 patients are lung adenocarcinoma and 553 patients are of lung squamous cell carcinoma. Now, what is my another question will be that this is the expression value of this gene in all the samples. Now I'm interested to stratify the patients on the different conditions, basically. Okay. The gene on the two different conditions. What are the conditions? One condition is what? Lung adenocarcinoma, and another condition is what? Squamous cell carcinoma. So I'll be interested to basically compare this gene in the two different conditions. Compare this gene in the two different conditions. So see. So for that, I have created a function here, basically. Okay. Sorry, guys. Extremely sorry. So what I'm doing here now basically is that sorry. Now you can see here that we are showing the description of this gene in the lung adenocarcinoma. Okay. Just a minute. Okay. Fine. Sorry. Yeah. So now you can see here that we are showing the expression value of this gene, the description of the gene in the two different conditions separately, LA and the LS, right? So you can see here now that the mean value in this condition is 7.1. Mean value of this in LS is 6.00, right? It means what? It has higher value in the LA condition. Standard deviation 2.22, standard deviation 2.44. 50th percentile 7.23, 5.80. Median is less than this. Maximum is 12.002, maximum is 12.40, right? Now suppose we want to understand this summary in form of visual, right? So what you can do now, for that you can make a plot. Can anybody tell me what plot will help us understand this information? Anybody? Hello guys. Guys, please interact, that will be good. हाँ मिताली, आप बताओगे क्या प्लॉट आप बना सकते हो इसके लिए? हेलो.
Yes, sir.
क्या प्लॉट हम बना सकते हैं इस डेटा को अंडरस्टैंड करने के लिए, इन दोनों को हम अलग अलग देखने के लिए?
So we can make bar graph and
bar graph is not a good plot here. Okay.
line
Jaiba would
graph,
like to answer. Yeah, batao.
box plot.
Yes. So box plot is the right answer, definitely. See. So now what I'll do here. So see, it is the box plot of the gene. It is the box plot, not the bar plot. Box plot of the gene across all the samples, across all the samples, no stratification. Now here I am showing the plot by stratification of the condition. Right? See. Now we can clearly see here that in the lung squamous cell carcinoma it is lower, in this it is higher. Now this is named as differential expression, guys. Right? भैया इसमें कम है इसमें ज्यादा है. अभी कितना ज्यादा है, लेकिन ये एक क्वेश्चन ऑफ इंटरेस्ट है. कम ज्यादा तो दिख रहा है, लेकिन कम ज्यादा क्या स्टैटिस्टिकली सिग्निफिकेंट है? और ये स्टैटिस्टिकली सिग्निफिकेंट का मीनिंग क्या होता है? व्हाट इज द मीनिंग ऑफ स्टैटिस्टिकल सिग्निफिकेंस? Right? These are the kind of the question that you need to ask being a mathematician or a statistician, basically, right? Now, this is through the box plot visualization. Now we'll be interested to ask how the values are distributed in the different across the different range, basically, right? So for that, we make use of a density plot here, guys. Now what you will find here basically is that in this range, that is 4 point something and 6 point something, the expression is maximum. It means what? Maximum number of patients are having the values between 4 and 6. This is the meaning of how to interpret this plot, basically. Okay, so this is named as modality. So this data has a bimodal nature. Okay, so wherever you have the peak is going to be called as a modal or the modal value, basically, right? So here the model value is near to the 6 and then again to the 8 point something. Okay. And we know this thing, like in our data, we have the two conditions, right? So it is great that density plot is also showing that bimodality is there. And this bimodality is coming from what? Due to the two different sample subtypes, the two different cluster of the patients, basically. Right? Now I want to show this with the help of on the two different conditions as well, basically. Now you clearly see that the expression in this blue one, that lung adenocarcinoma is higher as compared to the lung squamous cell carcinoma. Okay. Now, suppose if you're interested to show that there is one more statistical term in the field of data science, basically is that, and that is skewness. That ऐसा तो नहीं है कि लाइक मेजॉरिटी ऑफ पेशेंट में वैल्यूज एक पर्टिकुलर रीजन में है और बहुत ही कम पेशेंट में हाईली स्क्यूड है. So skewness is a degree of asymmetricity, basically. Degree of asymmetricity. So let me show you what is the meaning of skewness. Is the skin visible, guys? Can you see?
Yes, sir.
Good. So see here, guys. This is the meaning of skewness. Now this is a normal distribution. It is symmetrically distributed. This is a symmetrically distributed. Now this is positive skewed. This is positively skewed. Longer tail towards the right side. This is negative skewed. Longer tail towards the left side. आप ये ग्राफ देख के कहोगे क्या पढ़ रहे हैं फालतू की चीजें, but trust me, when you go for the modeling, these are the things which really matters. Okay, that if the data is highly skewed, you cannot do the analysis normally. You will have to make some data transformation techniques there, right? So these are the things that we need to understand in statistics, and that's why statistics plays such an important role here. Okay, so what is the degree of skewness and all that we are showing here, basically? And now you can see that like overall skewness of this particular gene we are showing here is negative. It means it's a negatively skewed data. Okay. Similarly, we have a other metric that is called kurtosis. So kurtosis basically means pickedness. Like this is named as pickedness. That is how well the peak of the data is, basically. Okay. So that is also negative here. So fine. So again, so this is negative skewed data. Kurtosis, let us forget for a minute. मैं kurtosis अभी वो कर दे रहा हूँ comment. See, okay. So overall skewness of this gene is this one, negative. Skewness of this gene for LA is negative. Skewness of एक सेकेंड रुक जाओ यार, यहाँ पर भी हमें ये हमने गलत दे दिया है. जीन हमारा है HIF3A. Fine. Oof. Fine. Now you can see here that the gene is negatively skewed in all the three conditions. Like overall skewness is also negative. Negatively skewed in LA also, negatively skewed in the this also. Okay. And now somebody asked a question here, like I guess Darshana had raised a question basically in the beginning that guys, whenever you are saying something, please unmute and ask, okay? Because I will not be able to see the chat, okay? See, so one question basically was asked by Darshana that how can dimensionality reduction techniques like PCA-TSNA help understand gene expression patterns? Great. So that is here, like, okay, so let me show you what is this. So guys, you are interested in basically that, see, based on the expression of the genes, how the different patients can be clustered together. Okay, how the patients can be clustered together. And this is how this, see, we can clearly see that we are able to see two different clusters of the patients, right? And this is a dimensionality reduction technique. What do we mean by dimensionality reduction? So let me answer you. We know that we have the limitation that we can only visualize two different dimensions on the either X or the Y. maximum of 3, x, y, and the z axis, right? But in the real world, especially in the field of biomedical sciences, we have the data which are dimensioned in thousands. Like we have 20,000 genes, okay, proteins, single cell, when we do, number of cells are very, very high, right? So you cannot visualize all the features, okay? So what we need to do basically is that we need to visualize the samples based on the features, which can be a latent representation of those features. So suppose we have 20,000 features. I need to find a feature which is going to be representative of those 20,000 features. A feature which is going to be representative of those 20,000 features, guys. Okay. And this is the meaning of dimensionality reduction. Okay. And this is how PC is one of those techniques. Okay? PC is one of those dimensionality reduction techniques, with the help of which we are trying to show the patients how the patients are present in the lower dimension space.
Sir, please repeat the PCA technique one.
PC is nothing but simply, it's a dimensionality reduction technique, which is going to help you lower down your dimension from very high dimension to the lower one. Because you have 20,000 dimensions, right? Gene expression has total 20,000 features. And you cannot visualize those 20,000 features, right? So what we do basically is that, what we do basically is that we lower down the dimensions which are going to be representative. Just a minute, okay? Can you see my screen, guys?
Yes.
Hello?
Yes, sir.
Yes.
Good.
Yes.
See, देखो ऐसा होता है, बहुत ही सिंपल है. आपका जो डेटा है उसमें आपका सैंपल्स हैं. सपोज 4 सैंपल्स है, ठीक है? ओरिजिनल डेटा में कितने जीन्स है? G1, G2, gene 20,000, right? Now, we know that these 20,000 we cannot visualize because maximum आप क्या दिखाओगे? यहाँ पे X, यहाँ पे X2, यहाँ पे X1. मैक्सिमम होगा तो आप X3 दिखा दोगे. अब ये 20,000, क्या आप ये 20,000 दिखा सकते हो किसी भी तरीके से? बताओ मुझे दर्शनों. Like it is not there. But also very ask me to explain again. Can you represent these 20,000 features directly here? Bolo?
No, sir.
No. So what we need to do basically is that we need to find some latent representation. Latent basically means the features which will be a representative of these features, but in a hidden, in a different dimension, in a different space. It is going to be in a different space basically. Okay. So now we are going to find a representation which is named as PC1, which is going to be something like C. Alpha 1 G1 plus alpha 2 alpha 2 G2 plus alpha 3 G3 and so on till alpha n G20K. Okay. So like this, we'll find some latent representation. Now this PC1 is making use of all the genes. It's making use of all the genes. Similarly, we will find PC2. We will find PC3 and so on. We find PCN basically. Okay. We find number of PCs. But the exact number of PCs and all that I'm not going to touch here because that is pure mathematics. Then you will अब मुझे कोसोगे कहोगे भाई कुछ समझ में नहीं आया, ठीक है? तो उसको हम skip कर रहे हैं. We find a number of PCs and PCs का rule होता है that they are arranged in the order of decreasing variance basically. तो PC1 जो होगा आपका maximum variance explain कर रहा है डेटा का, followed by PC2, followed by PC3 and so on. So what we do then basically is that now you can PC1 and PC2 you can easily visualize only two. So what you'll do now? You will plot PC1 here, PC2 there, and you will plot your samples here. क्या दिक्कत है? Got it? So this is why we perform PCA basically to lower down the dimension and visualize our samples in the lower dimensions. Got it?
Yes,
Yes,
sir.
sir.
Thank you.
मैथ्स मत पूछना, बिकॉज इसमें नहीं आ पाएगी आपको, मैं एक्सप्लेन नहीं कर पाऊंगा अभी आपको, ठीक है?
Sir, I have a quick question.
हाँ जी, पहले आप नाम बता दो फिर क्वेश्चन पूछना.
Zeba, sir.
या प्लीज गो अहेड.
Sir, actually, I wanted to ask you, like the PCA, we use two kind of methods, right? Linear and non-linear. So I just wanted to know about these things. Like PCA is linear,
PCA is a
right?
linear method basically, Java.
Yeah. So, sir, where we can apply non-linear, and what is the basic difference between these two?
Good. So that is a very nice question basically. Okay. That when to apply linear method, when not to apply linear method, right? This is the question. Am I right?
Yes, sir.
Good. Fine. So see, basically, like we know that like it first comes from the domain understanding of the subject, basically Java. Okay. ऐसा कोई हार्ड एंड फास्ट रूल नहीं है कि ये डेटा होगा तो आप डायरेक्टली इस पे पीसीए लगाओ और टीएससी लगाओ. ऐसा कोई हार्ड एंड फास्ट रूल नहीं है. अगर पीसीए लगाया तो टीएससी नहीं लगा सकते, टीएससी लगाया तो आप पीसी नहीं लगा सकते. ठीक है, पहला थिंग ये, आप कुछ भी अप्लाई कर सकते हो. बोथ आर मेड फॉर द पर्पस ऑफ dimensionality reduction. Similarly, we have the UMAP also. It is also a non-linear method basically. Another point is that whenever from the domain knowledge, you know that this data do not follow linear in nature, is not linear in nature. We should always go for non-linear methods basically. Okay, and we know that gene data sets are also not linear in nature. So we should definitely go for the TSNE method first of all. Okay. Second thing is that explainability. TSNE, you will not be able to give much explanation basically. It is non-linear in basically, but you are not able to get the idea about the variance. You are not able to get the idea about the different features. Okay, because PCA is a linear method. So you are giving the different loadings to the different features. So you can get an idea about the different feature and their importance as well through PCA. But the TSNE, you will not be able to get that. Because it's a non-linear dimensionality reduction technique where you just try to maintain the topological distances. Okay, that is how the samples are closer in the higher dimension, and we try to balance those relative distance in the lower dimension as well. Okay, so explainability and only explainability is the main factor basically. Java, if you want some explainability to be there, go for PCA. If you just want to visualize, you can go for the TSNE as well. Okay, you can make use of either of them, but the complete understanding comes from the domain understanding. ऐसा कोई hard and fast rule नहीं है कि आप एक लगाओगे एक नहीं लगा सकते given data में basically. Got it?
Yes, sir. Thank you.
Good. So here what you can see now basically is that, as I was telling you, that PCs are arranged in the decreasing order of variance basically, right? So here you can see that PC1 is explaining 19.2% of the variance of the complete data. And PC2 is explaining 7.17% of the variance. So total 26%. So out of the 100% variance, only first two PCs are explaining 19 to 26% of the variance, which is not bad in order to visualize, right? And then with the help of these two components, we are visualizing our samples. So these are the samples basically. Okay? They are the different samples of the different conditions, lung adenocarcinoma and squamous cell carcinoma. And we clearly see that these patients are present very separately from each other. Okay. But now see a question. The question is that I was telling you here that these two genes, this gene has different level of expression in these two conditions. Now the question is, what is this difference statistically significant or not? So Jabba, can you answer this? Like which statistical method can we make use of in order to answer this question? Is this difference statistically significant or not?
Sir, which statistical method we can use
Yes.
in order to see the difference between these two?
Yes. Is it statistically significant or not?
Sir, t-test?
Yes, absolutely. Okay, so we can make use of a t-test, guys. Okay, so but again, now you need to tell me, Java, here that when can we make use of t-test?
Sir, if the data is distributed normally.
Great. And then other?
Sir, if data is not normally distributed, then we have to use which are non-parametric methods like Wilcoxon, I think.
Awesome. Great. Yes.
Thank you.
So that one you have told that like data should be normally distributed. So now the other question of mine is that like, can you tell
There
me
is
like which
number of observation is more
great.
than
So Garima is
30.
also the right answer. The another assumption is that number of observation is small. Yes. Definitely it's the right answer, Garima. Great. Now my another question is that any of you can tell me basically like how to check if the data is normally distributed or not. बताओ जैबा कैसे चेक करोगे डेटा नॉर्मली डिस्ट्रीब्यूटेड है कि नहीं है?
Sir, we have to apply some statistical tests like Shapiro-Wilk, I think.
गुड. ये आपने यहाँ से पढ़ा है कि आपको पता था सच बताना?
Sir, थोड़ा थोड़ा knowledge है.
चलो गुड. दैट्स ग्रेट. यस. So we can go for the
Actually, I'm
Shapiro-Wilk.
also interested in data science. That's why I also look these things on my own
Good.
and try to learn on my own. But I think like some expertise advice I require.
Good, good. So हम इसको करेंगे बात, ठीक है?
Yes, sir, sure.
So see guys. So this is the pipeline that you need to follow basically. Okay, that we have the gene name. Then we need to perform Shapiro-Wilkinson test. So Shapiro-Wilkinson test is a test which will tell you either the data is normally distributed or not. If the data is normally distributed, then you can go for the t-test. If it is not, then you can go for non-parametric test. Okay. So I'm applying here Shapiro-Wilkinson test. Now you can see here that Shapiro-Wilkinson test for HIF3A, p-value is 1.8 into 1e minus 6. Does not appear to be normally distributed. It is saying that data does not appear to be normally distributed. Okay. Then we are applying the test in HIF3 in lung adenocarcinoma. And here also the p-value is less than 0.05. Here also the p-value is less than 0.05. It means that the data is not suitable for performing t-test. Okay. So then what you can do basically is that you can apply any non-parametric test method. Okay. So this is the complete pipeline steps that I'm telling you. Like this is the thing that we need to follow being a data scientist. Okay. But yes, according to Shapiro, it is not normally distributed, but we can still apply t-test. Why? Because the sample size is above 30. And we will write the assumption here of this CLT theorem, central limit theorem. We'll state that based on CLT assumptions, we are stating that because of the high number of sample size, data will follow the sampling of the data will follow the normal distribution. For that, we are applying t-test. So you can still apply t-test basically. Also, we can make use of a plot that is named as QQ plot here. Okay, guys. So you can make a QQ plot basically to see how the data is normally distributed or not. So see, the data is normally distributed in this range from here to here. And here the data starts violating the normal distribution rule a little bit. Okay. So if the data basically, okay, you find a straight line, it means a good fit for the normal distribution. Okay. If it is not following on a straight line, then it is meaning not normal distribution. Here you have more violations and more violations basically. तो अगर हम सिर्फ इतना रीजन लेंगे यहां से लेके यहां तक और डेटा नॉर्मल डिस्ट्रीब्यूशन आएगा, ठीक है? Okay. So now I'm interested in basically to see what is the level of expression of change in the two different conditions, right? In the LA and the LS basically. Okay. So, and this is named as log fold change, guys. Okay. So fold change is this much 1.10 basically. Okay. And the next I'm not telling here because that is a part of the next webinar that I will take. That is predictive modeling and the differential expression. So till here only I will cover today. Okay. And now I'm open to questions or from the questions that you have. First, no, fine. But before that, let me show you something, guys. Okay. So hope I have not bored you guys. Okay. So I've left correlation and all that I will cover in the coming webinar basically. Okay. So please let me know if your screen is visible, guys. Okay. Is my screen visible?
Yes, sir.
Good. So this I have left, not a big deal. Great. So, yeah, guys. So last thing basically is that, guys, see. So these are the things that I've taught you basically today. Very important, and this is just the beginning of the field of data science. Okay, and the field of biomedical data sciences basically. Okay, so we are recently going to launch a course basically named as Omics Data Science. Okay, which will begin from the 15th or 20th of April this month only. Okay, so if you are interested in understanding and analyzing these kind of the data sets from the very basic depth, basically. This was the webinar, basically, where I have to give you the application and a little bit about the understanding. But in the course, we'll be covering all these things from the scratch. What we'll be covering? See, so we'll be covering basically preparatory module, where you will be learned all the biostats which are required, then the linear algebra of the different which are required in the data science, then differential and integral calculus and EDA part in detail. Basically. This is the preparatory module that you will learn. Module 2 will be teaching you complete machine learning, basically. Okay. So in the next webinar, where I'll be covering predictive modeling, basically. So that there you will get to know supervised learning. Like what is supervised learning? What are the different methods that we get to know? What is predictive modeling, diagnostication, prognostication, and all. Then unsupervised learning and deep learning fundamentals. And the last module, which will be the omics data analysis, basically, where you will get to understand in detail and the depth how to analyze genomics data, microarray data, multi-omics signature discovery, survival data modeling, and your this spatial transcriptomics and other different kind of the analysis, basically. Okay. Then definitely like GWAS and all also. Okay. So I will highly recommend, like, if you guys are really interested, feel free to like register for this course because this will be launching soon by the end of this month, most probably. Okay. And if you will attend all the coming webinars, also 4 webinars, then you will be able to get 20% discount, but you will have to attend all the webinars. Okay. And also I'll be giving some assignments, so which will be required to do. Okay. So this is us about, yeah, can you pardon basically?
Sorry, sir, I didn't get you.
Okay, so fine. So this is about the company basically. Like I did not tell you a little bit anything about the company basically. So we are a private limited company named as Brass Education Private Limited or aka TechMedBuddy. So far we have taught more than 1,000+ students, conducted a number of webinars and seminar basically, and the offline workshops in universities like JNU, Amity, DU, then Mumbai, Pune, and number of Bangalore. So number of different, not Bangalore basically, yeah. So we have conducted a number of offline and the online webinars and the workshops basically. And this course will be taught not by me only, like other than me, there will be 2 more faculties to teach you this particular course. Okay. And trust me, by the end of this course, you will be eligible for any kind of the job in the field of biomedical data science in market. Okay? Not any kind of job, but omics data science related ones basically. Okay. So yes, this is all from my end. So let me stop the recording.